Now, furthering the discussion regarding the monoclonal antibodies, in this module we will discuss how the monoclonal antibodies can be produced, how they are used, what are the animals which are there we are choosing for the production of these antibodies. Now, monoclonal antibodies, as we have discussed earlier, they are produced by the animal, by immunization of animals, different size of animals. Now here the mice are usually the animal of choice for the monoclonal antibody production. The mice, because they are of small size, they are economical to grow, they are economical to feed, and economical to keep under the known or the defined conditions. So, bulb C, that is unusual mouse strain, that is used for the monoclonal antibody production. This is a mouse strain which is specially used for the production of monoclonal antibodies. Now, again, mice are immunized usually 3 or 4 times over the course of 3 to 4 months. So you see 3 to 4 months it takes by the intraperitoneal route using antigen mixed with adjuvant. That the adjuvant are the compound which reduce the antigen quantity. The second is in the intraperitoneal route mean that they are injected in the intraperitoneal cavity of the mouse 3 to 4 months repeatedly. Now here you see there is a route which is depicted in this picture, that how we can make the monoclonal antibody production, where there is immunization, where there are the cells which is fusion with the antibody G, and further the test is carried out by ELISA. If the test is positive, then it is bled and the antibody is separated. But further, if it is not, again the cycle goes on till we get a titer which is actually required by the antigen production. Now, once the mice are sufficiently immune, they are left for 2 to 3 months, as said earlier, to rest. After 2 to 3 months, they are just fed and no immunization is done so that the antibodies are produced there. Mice are after that sacrificed and the spleens, they are removed from the mice. Now, single spleen will provide sufficient cells for 2 to 3 cell fusion, because mind you that we have to make a cell fusion after we get the spleen of the mice where it is antibody is accumulated. Now, how we extract the spleen cell? That is, the extract the mouse spleen cells which contain beta lymphocytes and culture them in the lab. We extract the spleen cell, culture them in the lab on the petri dish or in the tube, and then the extract mouse tumor cells which will grow continuously and culture them later on. That is, we will culture these cells again and again and again in the lab. Mixed spleen cells and the tumor cells which we get on the same plate. So we put the tumor cells and the spleen cells on the same plate in the lab, in the culture, and then add polyethylene glycol. Now, what is the function of polyethylene glycol? Is that this causes some beta lymphocyte to fuse with the tumor cells to produce a hybrid cell, which is called hybridoma. So see, now we are preparing the hybridoma. That we took those spleen cells, grow them time and again in the lab. Then we took the tumor cells, put the tumor cells on the petri dish or on a dish, and added the polyethylene glycol, which causes some beta lymphocyte cells which are there to fuse with the tumor cell, and that tumor cell is called a hybridoma. Then grow the cells under conditions that allow only hybridoma cells to survive. Then we have to take those mixture and put them or grow them on the condition where hybridoma cells, they can survive only. Now, after the hybridoma cells have grown in sufficient quantity, we will extract the cell culture and separate those hybridomas from the other cells. Test the medium again around each cell for the specific antibody of interest. That is the antibody, whatever antibody we want to form, that is the antibody or immunoglobulin Y, B, A, or whatsoever. So we will test that for that. So in this cartoon, you can see that there are different stages which are being indicated. That the mouse, it is first immunized, then the cells are taken. Here there are the tissue cells, then there is a fusion which takes place, and eventually we get the hybridoma which is prepared there, and from that we extract the monoclonal antibody in concentration, and that we get. Now, we culture the cells as shown in the cartoon, then culture the cells making the desired antibody and use as needed. Now, mind you that this antibody which we have prepared can be kept for long time under certain conditions and can be used appropriately whenever time arises.